Hi, welcome again to Propel's video series, where we highlight innovative research into the causes of Crohn's disease, as well as treatments and novel diagnostics. Today, we are very excited to have from London Dr. James Lee from the Crick Institute. Angela Clark, one of the members, of course, of Propel, will be interviewing Dr. Lee today. So take it away, Angela.
Thanks, Annabelle. I'm going to start with going over Dr. Lee's impressive bio, and then we'll launch into our questions. Dr. James Lee is a clinician scientist with clinical expertise in inflammatory bowel disease, or IBD. He seeks to better understand the biology of immune-mediated disease and translating that knowledge for patient benefit. Dr. Lee completed medical training at the University of Oxford in 2004 and his PhD at the University of Cambridge. He completed clinical training in gastroenterology as a clinical lecturer at the University of Cambridge before being awarded a Wellcome Trust Intermediate Clinical Fellowship in 2015. He spent 2 years of this award at Harvard University before returning to the University of Cambridge in 2018 to establish a research group at the newly opened Cambridge Institute for Therapeutic Immunology and Infectious Disease. He joined the Francis Crick Institute in London as a clinical scientist group leader in 2021. Dr. Lee not only seeks understanding of disease mechanisms, he is also interested in translating his research for clinical benefit, helping convert initial discoveries from his PhD into a prognostic biomarker that has since been validated, licensed, and made available for patients. He has published over 50 research papers, including first senior author papers in Cell, Nature Genetics, Journal of Clinical Investigation, Gut, and EMBO Molecular Medicine, and co-authored papers in journals including Nature, Cell, Nature Immunology, and Journal of Experimental Medicine. His work has provided insights into why patients with the same disease often experience different outcomes, including the discovery of a genetic contribution to prognosis that is distinct from the contribution to disease susceptibility. More recently, his group has developed approaches to leveraging genetic associations, providing insights into disease mechanisms. Welcome, Dr. Lee.
Thank you very much for having me.
We're first going to start with a groundbreaking study from your team that was published in Nature just last month that really set the IBD community abuzz. You generated international headlines, and the study really uncovered a major genetic mechanism behind IBD, and it was entitled "The Disease-Associated Gene Desert that Directs Macrophage Inflammation Through ETS2." So help us dissect this study to start off with. It seems this discovery centers on the role of a gene called ETS2 in IBD. Was this gene previously known, and how, in an overall sense, how is it a factor in IBD from what you found?
So thanks very much, Angela. So the short answer to your question is no, we didn't really know about this gene. We know a lot about disease genetics in IBD. So we know lots of regions in our DNA where something can go wrong and it contributes to why people get Crohn's disease and ulcerative colitis. The problem is that while we know where those bits of DNA are, so we can sort of pinpoint them, we often don't know exactly what is going wrong at these regions. So in fact, we've known about a particular genetic association on one of our chromosomes, chromosome 21, for about 15 years. And this region has not only been linked to Crohn's and ulcerative colitis, it's also been linked to a whole range of other inflammatory diseases. And it's been a challenge to try and figure out what's going on at that region, because actually where the sort of pinpoint is, where the disease risk is, actually sits in a big bit of DNA where there are no genes. So often when you do genetic studies, you find hits near to particular genes, and it's very easy to then come up with an idea as to what may or may not be happening. But when you find a region that's, as it describes, it's a gene desert, there is nothing there, sometimes it's very hard to then figure out exactly what might be going on. So for a few years now, my lab has been really interested in trying to unpick what might be going on at some of these gene deserts. And we kind of realized quite early on that a lot of the time, although we don't think there's anything there in terms of genes, what there often is there are what are called enhancers. And enhancers are a bit like volume dials for genes. So they turn up or turn down the amount of a nearby gene and how much of that gene is being made. So in many ways, they're the critical control elements within our DNA. And the interesting thing about enhancers is that they work differently in different cells. So you can have a bit of DNA that's a strong enhancer in one cell type, does nothing out at all in another cell type. So we started to study this region, and we quickly realized that actually within this gene desert, there was a very strong enhancer, and that enhancer only functioned in a type of immune cell called macrophages. And we know that macrophages are really important in IBD. One of the biggest changes you see in the intestine in patients with Crohn's is that they have this huge influx of inflammatory macrophages. And the other reason we know they're really important is that many of the inflammatory chemicals that these cells produce are the things that are targeted by some of our best treatments, things like anti-TNF or anti-IL-23. So what we ended up discovering was that this gene, ETS2, which is the gene that's regulated by this volume dial, and it took a bit of time to figure out what that was, no one really knew what it did in macrophages. And what we figured out was that actually it turns out to be the gene that sits right at the very top of the pyramid. So if you turn it on, it turns on all the inflammatory functions in macrophages. And if you turn it off, it turns off all the inflammatory functions of macrophages. And in fact, strikingly, you can take sort of resting, non-activated, completely healthy macrophages, and you can just turn up the amount of ETS2, and those macrophages end up resembling the macrophages that we find in the gut of patients with active Crohn's disease. So we think what we've sort of stumbled on by starting with this gene desert is a pathway and a gene that is absolutely central to the inflammation that patients get with IBD. And then that in turn gives you something that you can think about, well, could we now go in and actually intervene therapeutically instead of targeting one or other of the inflammatory chemicals that are right at the bottom of this big cascade? What about targeting the thing right at the top that's actually switching everything on in the first place?
That's very interesting. How did you, I think you talk somewhat in your paper about how you went about finding this and the process you went through. Can you tell us more about that?
So yes, I can. So we found that it was ETS2 that was being regulated through a whole load of different complementary sort of methods. Actually, one of the things that we've been able to do, we've optimized this in the lab, is actually being able to delete very small bits of DNA from cells using CRISPR. And actually, what we could do was actually remove the bit where the enhancer was actually in macrophages. And the nice thing about that is if you take out the thing that turns up nearby genes, if you remove it, then you can see which of the genes doesn't turn up anymore. And that's exactly the experiment we did. So we showed that actually if you physically delete the enhancer altogether, the only gene that is affected by this is ETS2, and ETS2 then fails to go up when macrophages get activated. So we use that. We use different other methods. Sometimes people, when they see schematics of DNA, they like to think of it as a long sort of straight line, whereas in actual fact, it's much more like a bowl of spaghetti where bits from over here might be physically touching bits from over here. And again, one of the things we could show is that you can physically show that this gene desert, although it sits a very long way away from where the ETS2 gene is, actually within cells, these two things loop together. So this is directly regulating the ETS2 gene. So that's how we found it was ETS2. And then, as I say, through experiments where we either could remove it or overexpress it, all done in human, primary macrophages, we found out that it's absolutely central to these inflammatory processes, many of which are already known to be, important contributors to the pathology we see in Crohn's.
Now, you mentioned the use of CRISPR for the laypersons watching. Can you explain more about the role of that and how it was used?
Yeah, so CRISPR's been something we've known about for a little while, and what it's enabled us to do. So CRISPR is essentially, it's something that can cut bits of DNA, and it doesn't just cut it randomly. You can actually design something called a guide RNA that will take the CRISPR machinery to a particular bit of DNA. So the best way to describe this, it's a bit like the sort of find function. If you're looking on a web page or on a document, you can type in the word you want, and sure enough, it will take you to the bit of the document where that word is. So CRISPR works in a similar way. We can give it the sequence that tells it where to go, and once it gets to that site, it will then cut the DNA at that site. Now, what that means we can do is we can actually designed these guide RNAs that sit on either side of the enhancer. And that means that you'll take 2 bits of the CRISPR machinery, and when it cuts, those bits will be removed. The DNA naturally repairs itself, but when it repairs itself, it often has had that bit excised from it. And that means you can then figure out in cells that were otherwise fine, you haven't done anything abnormal to them, you can then figure out what the function of that bit of DNA was. And in the same way, you can actually then knock out genes. So we were able to get monocytes and introduce a mutation into the ETS2 gene that meant it didn't work anymore. And that then enables us to say, well, okay, if you think about all the important functions of macrophages, which of those are affected when you don't have ETS2 anymore?
Just amazing work that you all are doing. How does ETS2 compare to other inflammatory regulators in our system?
So we've known for a long time that there's lots of genes that have been implicated in inflammation, and there's lots of genes that are known to amplify inflammatory responses. So we like to think of it in sort of 2 ways. If you remove a gene and then inflammation goes away, then there's lots of ways in which that might work. It might sit within an inflammatory pathway, it's somewhere in the middle, and you take it out and the inflammatory pathway can't work anymore. So that's completely possible. And that tells us that it's important and it's essential for inflammation. It doesn't mean that it would be sufficient to turn inflammation on just by itself, though. And there's lots of genes that fall into that category of being important in inflammatory processes. And there's lots of them, even things like TNF and things like that would all fall into that category. What's really unusual, though, is when you have a gene that is sufficient to induce that as well. So now we think about the opposite. We're not removing it, but we're going to overexpress it. And if you overexpress, ETS2 in a macrophage, everything switches on, which is really unusual. And this is the striking result, really, when we were doing this result, was realizing that just simply turning on this one gene took resting, non-activated macrophages and made them look just like they've come out of the gut of patients with Crohn's disease. So ETS2 is, when you think, the master regulator of inflammation. So there's lots of other genes involved in inflammation. There's virtually no other genes involved in inflammation that have this ability to literally turn everything on just by themselves. And that's why we think this is really special, because we've not just found another inflammatory gene, we found the thing that actually is the orchestrator of the whole process. And that's why, if you imagine a situation, we know already that many patients with Crohn's disease might be given a biologic, and that biologic will work in some people, but it may not work in everybody. Or sometimes it works for a while and then stops working. And that's because just targeting one molecule isn't often enough to get rid of the inflammation that drives someone's Crohn's disease. So people are now starting to think, well, maybe we need to combine biologics or do things. And what we're still doing, though, is sort of trying to pick off the things right at the bottom. So we've known for a long time that actually if we could find the thing that was directing the whole process from the outset, that would be a much more effective drug target, because you'd have a sort of cascade effect whereby if you turn off the central controller, then none of the other stuff can get activated in the first place. And then you have effects across multiple different cytokines. So that's what we think we found. And that's why we're so excited about this, because we think we know how to turn it off as well.
Well, let me ask you about something a lot of us out in the general audience have long heard about, which is mutation of the NOD2 gene, and long known to play a role in some Crohn's cases. Does your discovery about ETS2 tell us any more about this? Is there anything?
The NOD2 is probably one of the most famous genes that has been linked to Crohn's disease. It's probably the biggest, at least from the strength of the genetic association, it is the biggest genetic association that we see. People have still spent a long time trying to figure out exactly what NOD2 does and in what cell types it does. The one thing that was really striking for us is that when we looked at the whole suite of genes that ETS2 regulates, we found that actually one of those genes is NOD2. So we think that ETS2, as well as controlling lots of different inflammatory functions that macrophages do, it probably also controls NOD2, which again positions this very much at the center of IBD, because it's not only regulating all the cytokines that we target with our therapies, it's probably also regulating some of the most important genes that we already know about.
So I have a question. So, because a lot of times you'll get, particularly in Asia, they don't necessarily have the NOD2, the ones who have had been diagnosed with Crohn's. So is it possible that this gene that you have discovered is the reason for the Crohn's disease in those patients, even if they do not have the NOD2 mutation?
So none of the genetic variations that we're talking about are necessary or sufficient to cause disease, and that's common of all sort of common genetic variation. It's absolutely possible that this is, and we know that this already, that this is an important contributor to IBD in Asian populations. Interesting, in Asian populations, there's another gene which is almost as strong as NOD2. It's not NOD2, and that is also a gene that people are starting to think about drugs against. And in fact, ETS2 regulates that as well. So again, I think what we found is the sort of the central controller of all of the problems that happen in macrophages in diseases like Crohn's disease. And we know that actually the genetic variation at this gene desert is incredibly common. And it's interesting why that's so uncommon. You think it shouldn't be common because obviously getting these diseases is not good for us. And that's probably another discussion. But we know that this pathway is basically overactive in almost everybody with Crohn's disease, and therefore provides a very compelling, drug target that may be relevant. You're not going to have to stratify people depending on whether they happen to carry the mutation or not, because about 95% of Crohn's patients will carry at least one or two copies of the mutated gene desert.
Let me go back to something you just said, and you point out in your paper, I believe, that you cite up to 70% in Westerners, as a matter of fact, having this ETS2 variant, as opposed to 95% in IBD. So can you comment more on that?
So that relates to something called the minor allele frequency. So we all have two copies of every chromosome. And so if you take the overall frequency of the allele, it's about 70%. If you actually work out the maths in terms of that, what that actually means is about 50% of patients will carry two copies of the mutated allele. And then about another sort of 40 or so percent of patients, just over 40% of patients, will carry one copy of the mutated allele. So put that together and you have nearly 90% or plus of people with IBD who will carry this. Now, quite a few healthy people will carry this as well. And this fits with the idea that there's none of these things in isolation is enough. You need to have hits in different pathways. You need an environmental trigger. The one thing we found was really interesting was that in a lot of the times where you find these genetic variants that are linked to disease, they are the minor allele. So in other words, they are uncommon in the population. And that sort of intrinsically makes sense, because if people get the variants that cause disease, you imagine over time these things would be selected out of a population just from sort of evolution. In this case, it's not, it's the major allele. So, and in sub-Saharan Africa or in the Far East or South Asia, the allele frequency is even higher. So the 95% minor allele frequency will mean that virtually everybody with IBD in those populations will carry one of these mutated variants. And the thing that we found in the paper is that the reason we as a sort of human species have, evolved to live with this mutation is that it actually can be beneficial if people get bacterial infection, particularly very early within the first few hours of seeing a bacteria. And that kind of makes sense because the gene ramps up your macrophages and makes them more inflammatory. Of course, that could be a good thing if you've just been exposed to a bacteria and there were no antibiotics around. So hence, carrying this variant may have been protected before we had antibiotics. Now, obviously, we have antibiotics, but we're now seeing that those variants like this that protect us in previous, generations gone by are actually now making us have an immune system that's got a very fine hair trigger such that it doesn't take much to set everything off. So when we're thinking, this is also useful because when we're thinking about how would you design a drug to target this pathway, we know from the outset that we don't want to turn this pathway off altogether. That would probably be bad. It would probably leave people susceptible to infection. But it also means that we know that what we can probably do is just turn it down by 50%, something like that. And that may well be enough to switch off the inflammation very effectively in diseases like Crohn's disease. But hopefully will not be so much that actually it would leave people susceptible to infection. But obviously these things all need testing properly.
We've long heard that the genetic part of Crohn's disease. IBD is the smaller component. And in light of that, how does your discovery, or what weight should be given to this particular discovery in light of the part that environmental triggers may be playing? What's your opinion on that?
So we know, I mean, I think it's important to contextualize this, as you say. We still know that for any patient who comes into a clinic, the biggest risk factor for IBD actually is whether they have a family history or not. Mm-hmm. And so the way I tend to try and describe this to my patients is it's a bit like, it's a slightly macabre analogy, but it's a bit like thinking about firing a gun. So genetics loads the bullets into the gun, and then the environmental trigger pulls the trigger. So if you have a gun that has no loaded bullets in it, it doesn't matter whether you pull the trigger, it doesn't matter whether you see the environmental risk factor or not, you're not going to develop IBD. If, on the other hand, you have a fully loaded gun, but you never see the trigger to pull the trigger, that gun's not going to go off and you won't get IBD. So we know that you need both. So I don't think it's helpful to think about what's the most important bit, whether it's in genetics or environment, because actually we know that you need both of those things coming together in order for people to get IBD. Now, we've seen this huge increase in IBD and other autoimmune diseases over the last 50 years, and people have said, well, of course, the genetics hasn't changed, therefore it must be environment, and therefore the environment must be the most important thing. I think that logic is slightly flawed, because actually what that's telling us is we are so genetically predisposed to this that the slight change in the frequency of the environmental trigger is actually changing the overall instance of the disease. That doesn't undermine the fact there's a really important genetic component. It just tells us that actually, as a species, we have evolved, an immune system that has a very fine hair trigger, and it does not take much to make this go wrong.
That's a very apt analogy. Thank you. Do you think the ETS2 pathway is equally at play in Crohn's disease and ulcerative colitis?
Yes. So the evidence would suggest it is. We, as a lab, one of the things when we're selecting loci to work on, and of course, the beauty of genetics is we now have lots of loci we could potentially work on. When we're selecting which region to start thinking about, one of the things we try to do is identify those regions that not only increase your risk of both Crohn's disease and ulcerative colitis, but actually often increase your risk of lots of other diseases as well. And we do that for two reasons. First of all, it tells us that we're looking at really fundamentally important biology, because if a single bit of DNA being changed increases your risk of 5 diseases, there's something important going on at that site. The second thing, of course, is if you then find a pathway, as we did in this case, it means that thinking about developing a drug for that pathway isn't just going to be relevant for IBD. It might be relevant for 4 or 5 other diseases as well. So it sort of maximizes the potential that this could have for benefit of human health.
So you mentioned other diseases. What other inflammatory diseases do you see this ETS2 pathway?
Yeah. So we see it in ankylosing spondylitis, and of course that's also been linked to IBD. We see it, excitingly, we see it in primary sclerosing cholangitis, which at the moment we don't have any treatments for, and which 10% of IBD patients will also develop PSC. And in fact, something like 80% of PSC patients will get IBD. And in fact, in our study, one of the things we specifically did was go and look in the livers of patients who had PSC, and we can see that this pathway is not just present and switched on, but switched on exactly at the site of where the damage is being done at the bile duct cells. So we're pretty confident that this is directly contributing to the damage that people get with PSC. The final disease this is linked to is a disease called tachyacioarteritis, which is a rare type of inflammation that happens in large blood vessels and arteries.
In your study, you mentioned, talking about therapies, that we may be able to repurpose a drug called MEK inhibitors for that. Tell us why that is, how you came to that conclusion.
So we, having found that this pathway was there and switched on and clearly, doing something to contribute to, disease in IBD and other conditions, the obvious question was, is there a way to switch it off? Now, people have previously done structural studies on the ETS2 protein, And there doesn't appear to be a sort of intrinsic off switch within the protein. Some proteins have off switches, and then you can target a drug that essentially flips that off switch and the whole thing is straightforward. So ETS2 doesn't have that. So we then said, is there any evidence that we could find that there's some of the drugs that might already be used in people for other conditions might be acting, at least in part, through switching off ETS2? And so we went to a large database that has this sort of information, and we looked to see whether any of the changes that have been reported with over 6,000 different small molecules look like they might be acting through ETS2. And we found that there were several that looked like they might be. And when we grouped those according to their sort of known mechanism of action, the largest group that we found were these things called MEK inhibitors. Now, MEK inhibitors are interesting because they're already licensed for use in people. So they're used for cancer, they're used for conditions like neurofibromatosis. They're not known to be, anti-inflammatory, particularly they're not used for any inflammatory conditions. So we decided that that was worth testing out. So we got a MEK inhibitor and we looked at the inflammatory macrophages we can make in the lab. And if you put a MEK inhibitor onto those cells, it completely switches off the inflammation. And we can show that that process is almost certainly by switching off ETS2. And then because that was a slightly artificial system in lab, the other thing we did was we actually went and got colonic biopsies, so taken out a colonoscopy, from patients who had active flaring up IBD and who had not yet started any treatment. And those biopsies you can culture in the lab for up to sort of 24 hours. And in that time, they continue to release all the inflammatory chemicals that were being released when that bit of biopsy was part of the patient. And what we could do is actually add on a MEK inhibitor into that biopsy, into that culture. And we showed that that switched off the production of lots of inflammatory chemicals, including things like TNF, including things like R23, and a variety of other cytokines as well. So that's really exciting and that's really promising because it tells us we may have a way of targeting this pathway that could be very effective. The sort of the downside to this is that we know that MEK inhibitors have side effects. And for that reason, we know that they can't be taken for very extended periods of time at the moment. And the reason they have side effects is because MEK, the thing that's being targeted, actually does lots of other jobs in other tissues. So what we're now working on in the lab is to find a way to deliver a MEK inhibitor very specifically just to those inflammatory macrophages where you want to switch off ETS2. And we think we have a way of doing that, and we're testing this right now in the lab. And if that works, then actually what we will then have is the ability to turn off this inflammatory pathway just in the cells where it's going wrong without causing all the side effects that are known to associate with MEK inhibitors at the moment. So we don't have drugs that are ready for clinical trials right now, but I have someone in the lab we've made the prototype of this and we're testing this right now. And then the plan would be that if that all works, then we will actually be able to relatively quickly move forward into clinical trials, not least because MEK inhibitors have already gone through all the safety studies that they would need to go through in humans. And often that's a pretty big bottleneck in terms of drug development. So we hope we will have a significant head start.
So it sounds like you're really trying to modify it to really target it. Would these MEK inhibitor like drugs still inhibit the immune system? How do they compare to other IBD therapies out there right now?
Yes, so we don't know the answer to that yet because we will need to do that. The hope would be that by being very targeted to only get those inflammatory macrophages where ETS2 is causing problems, we would hope that you would not have such a profound effect on the rest of the immune system as some of the other drugs, things like steroids and things like that. But all of this is the sort of thing that we will need to test. And that's, as I say, why we don't have anything that's ready right now. But we hope to get to that stage pretty soon.
Right. Are there any other genetic pathways or factors that you all are exploring that could also play a significant role in the treatment of Crohn's right now?
Yeah, so we're doing quite a lot of work. That is what my lab is focused on. So I've got a whole team of people here in London who are specifically working on trying to uncover exactly what the genetics is telling us about IBD and what are the treatment options. So we know that macrophage inflammation is a major part of why people get IBD, but it's not the only part. So there's also contributions from other types of immune cells, things like T cells and other parts of the immune system and wider effects in the gut as well. So we are now actively working on a whole range of different projects using different approaches in order to have something that hopefully we'll do two things. We'll build up a much clearer idea of exactly why people are getting IBD. It's striking that in 2024, we still don't fully understand these diseases. And that's really evident because if you look at the drugs that are going into clinical development from big pharmaceutical companies, only about 10% of the drugs that go in at the beginning ever end up becoming approved treatments. And the vast majority of those that fail, so the 90% that fall by the wayside, they fail at what's called phase 2. Phase 2 is the bit where you figure out whether the drug actually makes people better or not. And so we have drugs that are going into clinical development that are targeting pathways, they're blocking cytokines or they're switching off molecules, and they do that very well. It's just that doing that doesn't actually make people better. And so we've known for some time that if your drug that goes into these studies is targeting a pathway that's actually supported by genetics, so it's directly involved in the disease, the chance of that drug working and becoming an effective therapy is massively higher. So it can go up, 4, 5, 6-fold higher than what we're doing at the moment. So we're using this as a means to not only understand the disease biology, but actually to potentially to identify drug targets that could provide the next wave of much more effective therapies for patients in the future.
Yeah, I think go ahead, Ella.
Right. So going back a little bit, with this treating this particular genetic, the ETS2, do you, for people who are flaring, and then for those who then go into remission, do you have an understanding of what's happening there? Like, why are they going into? Is this particular gene not doing what it was doing before?
So this gene is a gene that basically gets switched on at the slightest hint of any sort of information or exposure to anything that shouldn't be there. And as soon as that goes away, eventually, the gene will switch itself off. The problem we have is that because, when you get a tiny bit of inflammation, it self-perpetuates. So, your first cells might produce an inflammatory chemical, but that's enough to keep ETS2 going. And ETS2 then drives more inflammatory chemicals, and that's enough to keep the whole process going. So it kind of makes sense that you see these patients who have these flare-ups and it's just been grumbling for ages. And you think, well, why is it still going? Why isn't it? And actually, it's because there is a sort of self-perpetuating loop that's driving this. Now, if you could switch off ETS2, we're always looking for drugs that sort of break the sort of circuit, and that may well be a drug that could break the circuit, because if you do that and you do switch it off, the macrophages lose the ability to be inflammatory. And the macrophage inflammation, it doesn't just cause damage by itself. Macrophages produce chemicals that recruit in other immune cells. They present antigens to T cells, so they then set the T cells off as well. So again, if you get something that targets a very central process in a really important cell type, you may end up with a therapy that can very effectively switch off inflammation. And that's exactly what we're testing now.
Could it be something that would be temporary? I mean, in terms of taking the medication, that it would sort of be...
It's a great question. All of these things we have to kind of figure out. So we don't know the answer to that yet. I mean, that's the sort of thing that once we have compounds that are ready to test, it may well be something that could be temporary. Equally, it may well be something that is beneficial to have around just in case those macrophages start to become inflamed again, that you can switch them off very early. So again, we will figure that out if and when we get to the clinical trial stage.
What are your thoughts as to why patients with certain diseases like IBD can have such wildly different outcomes?
So that's a really good question. And it's something that I've worked on sort of before when I previously did my sort of PhD work in Cambridge. I think one of the things that we've tended to do as a community is we're very good at sort of siloing things. So, as a gastroenterologist, I almost exclusively look after IBD patients. I don't even see the other non-IBD related GI diseases because I've become so super specialist. And I think the danger of that approach is that you kind of miss the commonalities that occur between diseases. So we know that your question is a great question, but there will be someone in the world who's interested in MS who's asking exactly the same question. And there'll be someone in the world interested in rheumatoid arthritis who's asking exactly the same question. And I think for a long time, we as the community have thought a lot about what causes Crohn's and, why do people get UC? we've thought a bit less about, is there a layer of biology here that dictates for any autoimmune or inflammatory disease how frequently you flare up, or whether you're more likely to respond to the first treatment, yes or not. So I've done some previous work on that, and we found that there are indeed processes that control not whether you get the disease in the first place, but the frequency of the flare-ups that happen afterwards. So, there's a really important role for T cells in that process. And we've previously shown that one of the things that happens in some people is that those T cells kind of get, we call it exhaustion, but the T cells get essentially tired and worn out after a while, and they stop being able to mount flare-ups in the same way as they used to. And we can show that people who have more T cell exhaustion have far fewer flare-ups. So, their T cell exhaustion itself is not directly linked to why people get IBD in the first place, but it may be contributing to the disease course once they get it.
You've spoken previously about the distinction between the genetic contribution to prognosis or outcome and the genetic contribution to disease susceptibility, or how vulnerable one is to developing a disease. What are your thoughts on this subject?
Yes, so we've shown previously that actually the genetic contribution to disease course is different to the genetic contribution. I think that sort of ties in with the fact that, there does appear to be different biology. I think both are really useful to study. I think, frankly, we don't understand either well enough. At the moment, what my lab is principally focusing on are the pathways that go wrong when people develop Crohn's in the first place. If you're gonna ever even contemplate having a cure for something like Crohn's disease, what you really need is a proper mechanistic understanding of what goes wrong right at the beginning. And the genetics of susceptibility gives you the opportunity to do that. So that's why we're focusing specifically on that. But there's also a really important role for thinking about, once someone has Crohn's disease, can we understand why someone gets lots of flare-ups and why someone else might get hardly any?
I want to ask you about another landmark study that you co-authored last year, finding that changes indicative of Crohn's disease can appear in blood biomarkers up to 8 years before symptoms appear, which is remarkable. How can we use this information to ward off disease, and for those who are already symptomatic, perhaps help treat it? Is there a possibility of doing that, you think?
I think once someone has already symptomatic and has already effectively developed the disease, then I think that the utility of that finding is less. So the fact that we can show that there are changes in blood, in regular blood tests, that you can pick up, albeit you need a lot of patients to pick these up because they're very subtle changes, but they're real. We can see them happening in Crohn's up to 8 years before people are diagnosed. So this isn't a question of, someone's already got Crohn's and it's just been missed. And actually, this is something that goes on in that preclinical phase that leads up to the time at which the disease actually develops. So in terms of the utility of that, we looked in that study to see whether the changes were sufficient to be able to accurately predict who would get Crohn's, and they're not quite. They are a good start, but they probably need building on to make something that would be more useful. But there's lots of groups that are very interested in this sort of preclinical phase of Crohn's, because again, cure is one option for someone who's got Crohn's, but the other option that if you find someone who's really at high risk and who is on the way to developing IBD but maybe hasn't got it yet, is could you do something preventatively? So could you do something to ward off what will otherwise be, the inevitable development of IBD within the next sort of 5 years or so? And that's really where I think there's a lot of excitement, because, if you were to find people who were at risk and you could confirm that there was a good predictor that would identify those people who do have a very high risk, then there may be lifestyle changes. There may be changes in diet. There may be things like stopping smoking. There may even be, therapeutic options. There was a nice study done in rheumatoid arthritis not long ago from another group in London who have shown that actually intervening in the very high risk population can actually seem to reduce the development of disease in some people. So that's where I think a lot of this research is taking us. I think one of the big problems is we've never really understood much about that preclinical phase. And it is hard to study. You can't, design a study where we say we're going to recruit people who are going to get Crohn's, because you don't know who's going to get Crohn's. So people have been able to do this. There's some really nice work come out of New York, people like Jean-Fred Colombel, We've done a lot of work looking in some of the army studies where people recruited to the army have given samples many years, often before that they are eventually diagnosed with IBD, and you can go back and look at those samples. But the challenges with that is often you're not looking at very, very large populations. So the study we did was actually using a nationwide registry that's available in Denmark, where we ended up finding, 25,000 people who went on to develop IBD. And we could go back and look at their blood test results that had been taken up to 10 years before their diagnosis. And a study on that scale enabled us to find these very small changes that are definitely happening before patients actually develop the disease.
You're involved in a group called the UK and International IBD Genetics Consortia. How does this influence your research, and what collaborative efforts might be underway to translate these findings into patient care? Tell us about that.
So both of those groups are groups where we've actually been the groups who've actually found most of the genetic associations in IBD. So actually, from my perspective, this has been incredibly productive, because when I started as a scientist, we knew of only about 4 or 5, regions in our DNA that were contributing to getting IBD. We now know of nearly 400. So, and it's only been possible through large scale international collaboration, where we've been able to collect DNA from patients and have similar numbers of DNAs from healthy controls who don't have IBD, that we've then been able to look across people's genomes to understand where are the bits of the DNA where, in IBD patients, there's clearly a signal at one particular bit of DNA that's likely to be contributing to the disease. So that's been hugely helpful. As I said earlier, the genetics, finding, drugs that hit genetic pathways is a very powerful way of developing much more effective therapies. The challenge is that actually finding the bits of DNA where something important is going on has actually been not that difficult once you've actually got the samples. The real challenge now is translating that knowledge of where something is going wrong to an understanding of how it's going wrong. And that's the work that we now do in my lab at the Crick Institute.
Yeah, I was going to ask that about the ETS2 with the finding of the 8 years before symptoms happen. I'm assuming that you're looking at that and seeing whether it's starting to be activated, and does that activation increase as time goes on?
So we can't do that study based on the data that we have from the large nationwide cohort, because these are just regular blood tests that people have been having as part of their routine clinical care. They're not specialist tests to measure things like ETS2. And we still don't have samples from enough people that we could go back and do those sort of tests on. So I think it's a really interesting question. It's one at the moment we can't answer.
Any other questions, Annabelle?
No, I think this has been very, very interesting. And it explains a lot to people like us who, when you'll get you'll see the studies that they've implicated another gene, another 10 genes. It used to be 150 genes, and it's gone up to 200, and then it's gone up to 240. It just keeps getting bigger and bigger. And then it becomes, you begin to think that nothing is ever going to be solved, because it just, the enormity of this, the contribution of these particular genes just keep growing. And then it's sort of like, well, which one? So this is very helpful, because it does help to explain all of that maybe these other genes are downstream from this ETS2.
Yeah, so in fact, we show that that's exactly the case. So one of the things we did in the paper was, having mapped out what the ETS2 pathway was, because of course we didn't know what it was before, we went back and said, if we take all of the known hits, and at the time we did this, it was about 240 hits that have been associated with IBD, and we looked to see, are those enriched within the ETS2 pathway relative to other things that have come out, things like autophagy and TNF signaling or whatever. We ended up finding that the ETS2 pathway was more enriched for the genetics of IBD than anything that had been described before, including autophagy, including the intestinal epithelium role, including T-cell biology. So actually, we've not just found the pathway that is the central orchestrator of inflammation in macrophages. It looks like it's one of the most important pathways in contributing to getting Crohn's, and it makes a lot of sense of the genetics now.
And with the possible treatment, I'm assuming you wouldn't be able to do CRISPR or something like that with this nut, but
no, so we're basically trying to modify the MEK inhibitor in a way that would mean that it would only become activated when it gets into macrophage cells. So we're not thinking of doing CRISPR. There are some CRISPR therapeutics that are available for other conditions. We're not thinking of doing that at the moment. What we're trying to do is be able to specifically deliver that MEK inhibitor only to the macrophages that are, inflammatory in conditions like Crohn's disease, and not or somehow protect other cells in the body from actually being exposed to that.
Well, we're very grateful for all the work you all have been doing. You give the IBD community such hope. Please, I know you'll keep us and the rest of the world updated on what you find. I've been talking to Dr. James Lee. I'm Angela from Propel. Thank you for watching.
Thank you, Dr. Lee. Greatly appreciate this. And I echo what Angela just said. You do give us a lot of hope. So thank you.
You're very welcome.